Do you need this stand? Hi.
Everybody, thanks for coming the second week of seminars. First, it's an open announcement for grad student capital.
Hello, everyone. I'm Mackenzie. I am the graduate student council rep for bioengineering. I sent out a message yesterday or the day before, I don't really remember, detailing all the stuff from the graduate student council to highlight travel grants are open. So if you're traveling, please apply for a grant and get funding from the grad school to cover a part of your travel costs. And B, if you're a master's student or you're not candidacy, you notice that there was an athletic fee applied to your account, $200 and something a semester. We are trying to get feedback from a survey that's in that message in order to kind of draft a resolution to either decrease or prevent further increases to that athletic fee. So please go through that survey and give us feedback so we can take it to the higher level, try and get that fit. But yeah, there's also details of different events and stuff that are going on, free food, craft, stuff like that. So please take a look.
Thank you very much.
So today, we'll periodically.
Today we have Hossam Balaha with us from Dr. I'll give you an update on his work, his views with junior football. With that, I'll give Hossam the floor.
Good morning, everyone. This is Hossam Balaha. I'm a PhD candidate in the bioengineering department. This is my fourth year, for around to reach fourth year in this department. And I'm today presenting a transformative AI-driven framework designed to improve the assessment and outcomes of deceased donor liver transplants, which is a critical need for the organ shortage and variability in transplant success. My dissertation proposal is under the title Advancing Liver Transplantation Through Generative AI, Predicting Outcomes and Enhancing Viability Assessment of Deceased Donor Livers. This project is under supervision of Dr. Ayman Al-Bas. So as the title mentioned, we are dealing with the liver, and to understand the size of the problem in the medical field, we need to know the functionality first of the liver and what happens when it fails. The liver performs over 500 essential functions for the body, including detoxification of drugs and metabolic waste, protein synthesis such as clotting factors, metabolism of carbohydrates, fats, and hormones, and bile production for digestion and immune regulation. Unfortunately, when the liver fails, there is no machine that can fully replace it compared to other organs such as kidneys. Therefore, when the patient sees progress to end-stage liver disease, or in short ESLD, which can result from cirrhosis or alcoholic liver disease or NASH, the only cure for them is the liver transplantation. Without that, survival is often measured in months. The figure in front of you shows a 6-step progression of non-alcoholic liver, fatty liver disease, which starts by a normal liver and progresses to fat accumulation and fatty liver. In these 2 stages, they are easily reversible with interventions. And the 4th step, which is called NASH, where a harmful amount of fat accumulation exists in the liver, followed by the 5th step, which is NASH fibrosis, where inflammation is present. These 2 stages, 4th and 5th steps, can be reversed with intense interventions. And the final stage, which is called NASH cirrhosis, is irreversible as the liver cells die. So as mentioned, liver transplantation in this case is the only cure, and the primary source for the liver is DDLT, or deceased donor liver transplantation. And this case accounts for more than 90% of the liver transplants operations. As you see in the figure on the left in front of you, there are multiple steps and for the liver transplantation being followed by hospitals, and these are a set of protocols. One of these steps which we are working on in the histopathology study. From the histopathology perspective, determining the suitability of a deceased donor liver transplantation, or in short DDLT, involves comprehensive evaluation using multiple stains such as MT or methylstrichrome, and another stain called hematoxylin and eosin, or in short H&E. The H&E stain is a fundamental stain for assessing the general tissue structure and cellular morphology, allowing different pathologists to identify any significant abnormalities, inflammation, or any signs of disease in the donor liver. Meanwhile, in the MT stain or methylstrichrome, this stain provides detailed insights into the liver fibrotic changes by highlighting the collagen deposition, which is crucial for evaluating the extent of fibrosis or cirrhosis. By examining these 2 stains, H&E and MET, digitally, pathologists can achieve enhanced evaluation and visualization, and can employ advanced image analysis tools to quantify the fibrosis, assess steatosis, and detect any other microscopic features that could impact the graft function post-transplantation. Again, we mentioned in the words stains, slides, or sections. So what are those? To understand the idea and the full picture of it, let us introduce this flow from obtaining the biopsy from the patient until reaching the whole slide image scanning and applying analysis tools on it. So the figure in front of you shows these steps. It begins with the biopsy tissue, which is collected and fixed to preserve the structure before undergoing any processing, where it's dehydrated and embedded into paraffin wax to create a stable block. Then, next, thin sections are sliced from this block using a tool called, or a device called microtome, and mounted onto a glass slide. These slides are then automatically stained to enhance cellular details using different stains such as MT or H&T. These stained slides are then scanned using a whole slide technology or a scanner, converting them from a physical format to a digital format at different magnification levels, beginning from 2x or 4x for surveying large tissue areas, passing through to 40x or higher to get fine structural details, passing through intermediate magnification levels such as 10x or 20x for more detailed observation. So in digital histopathology, stains are chemical dyes used to highlight specific structures with biological tissues on a microscopic level, with the most common being, as mentioned, hematoxylin, adiosine, H&E, and MET. H&E stains the nuclei in colors blue or purple, and the cytoplasm in pink and red. And both of them, or H&E, aids to distinguish between abnormal and normal cells. Building on that, we need to mention that there is over than 12,000 patients are on the US wait list for the liver transplants, and this is annually, and only 9,000 or fewer transplants are performed each year. According to statistics, and in the figure on the right in front of you, in 2021, the wait list in the US for liver transplants is around 11,700, accounting for the second largest number after kidneys. Another challenge is the type of current assessment, which includes and relies on subjective and time-consuming methods such as manual Mason Stricom staining, or MET staining in short, and semi-quantitative fibrosis scoring, and those can lead to higher inter-observer variability and hence can lead to inconsistent decisions. We mentioned that the time is a challenge. To understand specifically how it's challenging, let us present the overall scenario. We have a brain death, and when there is a brain death for a patient or someone is declared, the donor liver is surgically recovered, placed in a special sterile fluid, which is a cold solution, and then transported to the recipient hospital, where surgeons must perform the transplant within the limited preservation window time to ensure the organ's viability. The recipient has to be ready for immediate surgery, as the liver can be preserved outside the body under strict conditions up to 12 hours. So, staining and digital histological assessment of the deceased donor liver must occur shortly after organ recovery to evaluate the fibrosis, inflammation, and overall graft quality before any transplantation. So this rapid evaluation is essential due to the limited window, as mentioned, approximately 12-hour viability window of the liver. Having recognized the importance of timely intervention and the complexity of liver transplantation process, we began identifying the different gaps in traditional methods from digital histopathology perspective, aiming to proceed and propose effective solutions to address them. These gaps can be summarized as follows. MET staining, the first point, is a labor-intensive, costly, is about or approximately 10x compared to H&E stain, and there is a delay in the decision-making. Staining and assessment can take up to 4 hours, and we are limited with the window 12 hours. Second, the fibrosis quantification methods rely on subjective scoring systems such as ISHAC or Metafer, and therefore they lack precision. And in some cases. The third point is a special differences between H&E and MET stained sections, as we are going to cover in the following slide. Fourth, there is differences between lab-to-lab staining and assessment, and this can affect the reproducibility of the decisions. And finally, the integration of clinical and histopathological data and markers is not applied in many cases. Thus, these inefficiencies hinder the timely intervention and decision-making, and hence can lead to an unstable balance between organ viability and patient survival. As mentioned, we are dealing with differences between the sections or the stained slices, and reflected on that, as you see in the figure in front of you, these are samples from our data set, and we are going to discuss the different gaps on them. The left figure shows different sizes between two applied and corresponding stains of the same patient, of the same organ, and as you see, their sizes are not uniform or not equal or even equal. The upper right figure shows how two tiles or patches or images of two corresponding stains, and as you interpret there are misalignments and differences in the information or the anatomical structure between both of them. Simply to understand the idea, when the doctor or the hospital tries to get H&E MT staining, they get the organ and proceed to follow the different steps of getting the whole slide image. And the procedure is followed as they get for a specific tissue or a specific slide, and they stain it with H&E, and the corresponding or the before or after slide is applied to MT stain. So for sure, there are some differences into the anatomical structure between these two stains. The lower right figure shows staining and scanner variability. The left column shows MT staining and the right column for MT staining. And as you see, for these two images, they are different in colors. And we are going to cover this gap in our methodology. So after understanding the idea behind staining and diagnosis, we began identifying the core ideas needed to address these discussed gaps. We realized that we need to develop a generative AI system that can basically generate virtual stained slides from only H&E stained slide, so we can get only one slide and generate any other slides or stains, automatically quantify the process and get away from the traditional manual methods. And third, predict the outcomes of the transplant. With that, this will reduce or eliminate the need for any physical staining, standardize the fibrosis assessment, and enable real-time or data-driven transplant decisions. Therefore, our solution uses deep learning to generate virtual MT images or slides, quantify fibrosis objectively, and predict transplant success, integrating all of that into one intelligent pipeline. So we set three specific aims for my dissertation or my proposal. The first specific aim is to generate virtual MT from H&E or to generate any stain from a single stain. Simply, we use H&E because it's less in cost and less in time and assessment. And we aim to reach 95% as a diagnostic reliability. The second specific aim is to automate the fibrosis quantification using AI, and we aim to reach 95% accuracy compared to the pathologist. The third specific aim to predict the transplant viability, and we also aim to reach 95% or more accuracy using the fused data and markers. We hypothesize that a generative AI system integrating the histopathological and the clinical markers can significantly improve the prediction of DTLT outcomes and post-transplant viability. All of these specific aims build on the last. So we generate the data, we extract the different markers and features, and predict the outcomes. All of them together form a closed-loop AI system for transplant optimization. Looking specifically for the first specific aim, which is generating augmented MET slides or any slides from a specific slide, which is H&E whole slide images with an accuracy of 95% or more. We used in our methods transformer-based GAN for stain translation. The input should be the H&E whole slide image, and the output should be MET slides or virtual MET slides. And we introduced the transformer as we are going to discuss, and this transformer uses a long-range dependency modeling to preserve the tissue structure. Our data set for validation is 35 patient samples with paired H&E and MET slides. And also we applied pathologist slide evaluation, and we used quantitative and qualitative metrics such as similarity index and mutual information. The second specific aim is to quantify the fibrosis in DTLT patients with more than 95% accuracy compared to the expert pathologists. And here, our cohort will include more than 30 patients, and we are going to compare our AI system with the pathologists. We will use transfer learning, including ResNet or any transfer learning models, and using simple models for robustness, and use fusion to preserve collagen fibers and cell boundaries. We will use different metrics, including ICC for reliability and blunt Altman for bias and detecting the limits, and the percentage of the agreement between our system and the pathologists. The third specific aim is to integrate the histopathological and clinical markers to predict transplant success, aiding to get 95% as accuracy. We will collect different inputs and markers, including histopathological data, including steatosis, necrosis, and fibrosis percentages, the donor markers such as cause of death, BMI, age, sex, so on, and the recipient information. And we are going to use different models, including random forest neural networks. And we will also introduce different explainability into our system to explain our decisions, not only getting a high performance matters, but also we need to explain why the model got those decisions. And we are going to report in our system the risk score for the graft failure, the rejection rate, and first-year survival. The second and third specific aims are still under development, but the first specific aim that we are going to discuss today is nearly to be completed. So talking specifically for the first specific aim in the methodology, as we mentioned, there is a gap that the stained slices are not aligned or are not similar because they take one from a specific tissue and another from a specific slide or the following slide. And for sure, there will be anatomical differences between both of them. So to cover this matter, we used a pipeline to align and deform these two slides so that both of them can be nearly aligned together. So we applied global alignment between the whole slide images, and then local alignment from the tile or patch perspective, and then applied freeform deformation, and this will correct any local tissue distortions, and then applied an improved version from Gibbs probability distribution to refine the clustering. And finally, we applied weighted fusion between different models to gain a more robust out. So we have a whole slide image, and to give you the idea, we have a whole slide image, and simply as an average, this slide or this whole slide image has a size of maybe 10K or 20K by 20K width and height, and introducing that into your computer as a one-shot will cause failure to your memory or your processing. So the idea is, instead of dealing with the whole slide image as a whole, you need to extract different patches at small levels or at small dimensions. So we started by extracting different tiles and the patches, and for sure, the whole slide image or the whole slide image doesn't contain information, but also contain background. So we put a criteria in our extraction process of the tiles. If there is a mutual information with more than 0.35 and the cosine similarity between these two slides with more than 0.75, this means that these two batches or these extracted two batches are similar or contain similar information. So we kept it if this passes this criteria and discarded it if it doesn't contain or didn't follow this criteria. So now we have different extracted batches from the corresponding H&E and MT slides. Then we started to gain a homography matrix so that we can use that homography matrix to make alignment. And simply, we applied global alignment from the whole slide perspective and the local alignment from the tile perspective. To get the global alignment, and as I mentioned, the whole slide image as a whole maybe can reach more than 10K or 20K in width and height. So we got something like thumbnail, and there's a reduced version or a reduced reflection from the whole slide image in a small dimension. So we extracted the thumbnail in 1000 by 1000 width and height, and then applied global alignment following this approach. We first applying a SIFT detector, and this is an algorithm to detect between two images. And here we detected key points between these two thumbnails. Then we applied a brute force matcher and matched these key points using the k-nearest neighbor algorithm with a distance of 2. Then we initialize a list to store the good matches between the two thumbnails and extracted the key points which are good matches between these two thumbnails. And then, based on these descriptors or these key points, we computed the homography matrix using an algorithm called RANSAC. And this homography matrix will be used later again for the local alignment. But for now, we used as a global alignment this homography matrix to align H and E thumbnail to MET thumbnail. But the output that will benefit us for the next step is the homography matrix. It's a 3x3 matrix that will be used or can be used for aligning images. Next, after extracting the batches and applying the criteria of keeping or discarding the corresponding batches, we apply the same algorithm, but instead of using SIFT, we used an algorithm called ORP, and it's better for small images. And we got another homography matrix. Now we got two homography matrices, one for global alignment, which resulted from the thumbnail perspective, and another from the local alignment, which resulted from batches. We multiplied both of them together and applied that resultant matrix to the batches. So now we have an idea from the global or the whole image and the local, which is the small tile. We applied all of that to all of the corresponding and extracted batches. The alignment only is not doesn't guarantee that the images will be aligned or deformed together, because alignment just swaps to the right or the left, or moving it up or down, just or resizing or applying shearing and so on. But the anatomical structure or the inner details itself will be fixed and addressed by the freeform deformation. So then we applied freeform deformation algorithm. And here, simply, the idea behind it, it gets blocks or small blocks inside the image and tries to find how to align each small block inside the tile to align or be deformed to the other image. And the generated output is called stream plot or the deformation stream plot. And as you see here, if you look to this arrow, for example, it says that I need to move that block or this small block into that direction to be aligned or deformed to the other block. So we did that with all of the images, and to ensure that before and after deformation guarantee that analytical structure are deformed together, we got the absolute difference between H and E and MET before and after. When the number of black pixels increases in the output of this black and white image, this means they are deformed because the difference will be 0. But when the number of white pixels or grayish level pixels increases, this means there is some differences between the pixels or the details in it. So before this picture was grayish, and after deformation, it became that most of the pixels are black. After that, we notice that each tile or each patch consists of 4 channels, RGB and alpha. As you know, most of the images contains just 3 images, 3 channels, RGB, red, green, and blue, to get the full picture. But here we got an alpha channel, which reflects the background or the brightness of the image. And we notice that in all of the images, it contains a constant value, which is 255 or white color. So we focus that we can change that alpha channel with more details to be provided to the model. So we saw that we can get rid of that alpha channel, which is constant and consistent to all of the images. It doesn't give any information to the system, and replace it with any other information. So our team focused to get something called clustering, and the clustering here to help the model to be able to distinguish between the cytoplasm and the nuclei and background. And we notice that, and from H&E staining, we have three main things: cytoplasm, nuclei, and background, three classes or three clusters. So we initialized our cluster with three clusters and got the map or these different clusters behind that algorithm. Then we applied refinement. Why? Because simply when you apply clustering, some pixels will be like outliers. You may find that some pixels inside a cluster is not related to that cluster, and the same for other clusters. So we needed to refine that clustering operation. So we applied an improved version from the Gibbs probability distribution, which is an algorithm, and we followed these mathematical steps, which is beyond our discussion here. But the input for that algorithm is the cluster or the initial cluster, and the output should be the refined cluster. We applied different iterations, as you find here in the figure on the right. We apply different iterations to refine the cluster and get rid of any outliers pixels. So, and overall for that algorithm, we have the input to that algorithm, which will be H&E patch and the initial cluster. And if you notice that there are some pixels here that should be refined, which we call here outliers, and the output will be the new cluster or the refined cluster after a set of operation or iterations. Now we have the alpha channel replaced by that cluster. We combined all of that together, RGB and the new improved cluster. Now we have full information that will be provided to our model. Our model should get the input to be H&E, which consists of the 4 channels as we mentioned, and get MT stained or the virtual MT. We used the original cycle GAN design and improved it. So simply in GAN or cycle GAN, there are 2 blocks, generator and discriminator. The generator simply creates virtual or fake data to fool the discriminator, while the discriminator acts as a judge, trying to tell the difference between the real and generator data, which is the virtual data. And these 2 networks compute in a zero-sum game. And simply the generator learns by getting feedback on how to improve the virtual output to be more realistic. And the discriminator try to getting better to identify the virtual or fake data. Here in our model, we got the encoder inside the generator and replaced it with a transformer. So we have an encoder here in the original design and replace it with a transformer encoder to preserve more deep and structured information from the given data. We replaced it with an encoder, so we called it a transformer GAN approach. We kept the discriminator as it is because it's rule only to judge whether it's fake or real. Then to judge the output and to get an idea if the output is similar to or dissimilar to the actual MT stain, we made or we gathered different metrics, including similarity metrics and dissimilarity metrics. Similarity metrics including mutual information, similarity index, cosine similarity, and so on. And the similarity metrics such as mean square error or perceptual hash, and so on. And we are aiming to increase the value of the similarity and decrease the value of the dissimilarity. We gathered all of this information to get the full idea, including texture information, including the spatial information, and so on. To train the model, any deep learning or any AI model contains a loss function that you need to decrease its value to gain a high performance. So the cycle GAN original loss function contains 3 blocks or 3 factors: the adversarial, the cycle, and identity. So adversarial loss, this ensures that the generated images are realistic and match the distribution of the target domain. The cycle consistency loss preserves the structural and semantic content of the input image throughout the translation. And without this, the generator could learn to map inputs to unrelated outputs, and this is still a full discriminator. Finally, the identity loss. This loss helps preserve the original image's color and texture when the input is already in the target domain. And by balancing all of these factors, the model will gain meaningful mapping between the different domains without one-to-one correspondence between the training images. So here we made a simple trick. In our first two configurations, we emphasized the role of the cycle GAN, and in another two configurations, we emphasized the role of identity. And later, we combined the output in a fused version. And we will discuss it in the next slide. But first, let's introduce the third and fourth configuration in our experimental design here. If you remember, we mentioned that there is a scanner variability. As you see in the figure on the left, there is a coloring differences between different slides, and hence we need to overcome this challenge. Simply, what we applied is normalization. And compared to other normalization techniques such as L2 normalization, max absolute, and so on, these techniques can be applied to simple images or images which is not related to histopathology. But in histopathology, there are some specific normalization techniques, such as what we used here is the Reinhardt normalization. This technique tries to normalize the coloring and picture between the different stains. So we have now two configurations, the first and second related to the original data, and the third and fourth related to the normalized version from the HMD. Now we have four versions or four models. We trained them and got the output, and based on that output, we fused the data. We got the majority fusion between it. So we hypothesized that if all of those models vote that the pixel should be blue or that pixel should be other color, then the fused version hypothetically should be in performance perspective, should get performance better than the other or the individual configurations. We applied these experiments on 12 gigabytes GPU as a personal computer, and then moved our experiments to the HPC at University of Louisville, which is a supercomputer. It includes more than 95 gigabytes in GPU. And we did that because each experiment of those take about 1 month to be completed, and we needed to reduce the time, aiming to get more results. So we moved to that server, and we decreased the time from 1 month to less than 2 weeks per experiment. And our implementation used Python and PyTorch package, and we use the similarity and similarity metrics as we discussed. We have 4 configurations, and to avoid any data leakage, we split the data from the patient perspective, not the type perspective, to avoid any data leakage. 90% for training and 20% for testing. And we used one epoch with, we used one batch size with 500 epochs. And these are our preliminary results in front of you. The top portion is the similarity matrix, and the lower portion is the dissimilarity matrix. And we applied our trials here 5 times. And as we mentioned, we used our PC or our computer, and each experiment took 1 month. So now we have 25 experiments, which took large time, maybe than 1.5 years to be completed. And as you see, we have the mean and standard deviation of each individual configuration and the fused version. And if you notice, for each metric, the fused version performed better than the individual or each individual experiment. And this validates our hypothesis that the fused version performed better than each individual configuration. And also we reported the statistical analysis, including box plot and surviving plots. And as you see in the fused box plot in the mutual information plot here, at there is a gap or a big gap between the fused version and each individual configuration, and this facilitates and emphasizes that the importance of fusion between different models is important. Quantitative results is not always important or is not always judgmental to be following the good way or the good rule. So we made also qualitative assessment. So we got outputs from our models. This is the fused version. And then we introduced the output to our collaborator in School of Medicine, Dr. Debson. And we asked if these two columns, which is the third and fourth columns, are generated correctly or not. And we got the feedback. And as you see in the figure here, this is the actual one, and this is the virtual one. And as you see, the virtual MET is following with more than 95 similarity with the actual or the actual MET state. So when we complete the project, our contribution should be getting a framework that can be applicable to other stains, and it can be like a blueprint on how I can transform the organ staining and transplant. And here we are using fusion between imaging and clinical markers. And also we are using virtual staining to reduce the cost and reduce the time. And here we are introducing from the transplant perspective, we are introducing end-to-end DTLT viability prediction system. And the further impact in eye medicine, it promotes transparency as we are going to introduce SHAP and Lime explainable AI frameworks and set different benchmarks and then demonstrates the feasibility of generative pathology. Beyond the lab, we have different key impacts. From the clinical perspective, it's faster, more accurate, and for the organ assessment, and hence lead to higher survival. From the economic perspective, we reduce retransplant and complications. We can reduce up to 100K per case, plus or minus. From the workforce perspective, we are training now students in pathology and transplantation. And finally, from the open science perspective, we are sharing codes, models, and data sets. And we acknowledge that we have partnership with Kentucky Organ Donor Affiliates, or CUDA, and University of Louisville Liver Transplant Center. With that said, we can summarize what we did in points. AI can replace MET staining, quantify the fibrosis, and predict outcomes, but not doctors or experts. And here our system achieved more than 95% in the first hypothesis, specific aim, sorry. And then we reduce the cost, the time, and subjectivity, and improve the organ utilization and patient survival. So as a final message, imagine a world where every donor liver is assessed in minutes, not hours. Within the AI system can see what human miss. And here we acknowledge that we are getting more data to improve our system from University of Florida in USA with the collaboration with Dr. Ahmed Nagla, and collaboration with Mansour University in Egypt with the collaboration with Dr. Khadija. And these are our grants and IRP. And the last one is affiliation of the acknowledgement for the cluster or the big computer in University of Florida. And thank you for listening, and I'm open to any questions. So we completed the first specific aim, and you are now on the way to complete the second and specific aim. So we got the stain or the virtual image stain, and we are going to combine all of that to generate the whole slide image. And the whole slide image contains for the fibrosis. So the next step is to get information about this fibrosis and get the percentage of this fibrosis, and get after that we are going to judge whether the plan is going to be applied to the patient or not based on the fibrosis and other markers that we are going
very informative. Patient comment on the training time. It's like a month to right to the training. How about the inference or operation time?
So simply we have large amount of data and training will take time. But the clearance time should be that because we are dealing with single second for a specific portion of the data. So theoretically, we can say that it can be
Usually this one is
theoretically for now, until we complete the whole system, we don't have the full time from
How well do you see the clinicians track the output?
So as I mentioned from in the slide, we generated in the fourth column in each bar the virtual entity stamp, and we introduced that into our collaborator and School of Medicine, Dr. Debson, and he decided that these images should be deleted again. These images are suitable or more than suitable, and based on that, we retried our experiment and performed another experiment to improve our system, and this should be our final output.
Thank you.
QR code written to for these someone to
Speak online on Blackboard for you to give off of your feedback on how you otherwise, see you next week for our engineering order. Fair enough.
little bit more housing within the study. Thank you.
Oh,
Oh, that's all right. Actually, just said.